Yes, good evening to all the participants. So, welcome to the day 10 session on immunoinformatics and reverse vaccinology. So, before stepping on to today's session topics, can we recap the last session topics? So, what exactly did we discuss in the last session? So, I'm giving you a kind of a clue. So, what exactly did we discuss on the last session? They come fast. The previous batches, before asking the questions, the participants already saved some text, and just like that, they ping so many messages. Uh, Interaction is somewhat good in your batch. Very good. Initiated from a participant, network pharmacology. We discussed on network pharmacology. Good. We have also discussed on system biology. Good. Then what else did we discuss? Network pharmacology. What is that? If we have one plant or multiple plants used to treat a particular disease, if we take the most important bioactive compounds from these plants, and if we use these bioactive compounds for screening through Lipinski rule of five, drug likeness filter, or through ADME properties, or even we can, after that, we can use a strategy to filter through docking, final by screening, screening through simulation. Okay, so we can start with bioactive compounds of plants, and then we end up with a base drug after doing all these drug design methods. Finally, instead of simply restricting with a single target receptor, we also go with the interacting receptors, that is, interacting proteins with a hub protein. And we also study the expression patterns, and we also go with the docking and simulation studies for that interacting proteins. So collectively, we call this as network pharmacology. It's a strategy, okay, to end with what are the most important genes and how about our compounds interacting with that particular gene coding proteins. And system biology, we have discussed something related to types of omics. It is a combination of study of types of omics, structural biology information, that is studying about the structure of genes, structure of proteins, and then we also link with the network biology, isn't it? So all these things together, we call it as systems biology. So I just mentioned a one word, and I was describing a lot. How about the thing? What else did we discuss? Can I ask you the questions? Then I think I can anticipate answers from your side. Give me examples for systems biology databases. So come with your answers. What are the examples we discussed in system biology databases? Just now I told types of omics, network biology, structural biology. So all these things we study in systems biology. Yeah, so many examples are there. Mention one by one. Can't recollect what happened. So many, yes, cake, but cake, we see genome databases. Genomics, we can go with the cake. And string, very good PPI interactions. Other than cake and string, what else do you remember? Do you remember the proteome databases? What are the proteome databases we have discussed in system biology? Yes, good. Uniprot. In Uniprot, we have a proteome. And then interactome databases are available. Metabolome databases are available. Yesterday, remember, I explained you something called a network analyst. And we have discussed on DigiNet. You remember this? These are the exclusive tools we discussed on yesterday, isn't it? DigiNet is related to the disease gene network. Network analyst, gene expression networks. We can link with R programming, Python programming, Perl programming in these databases. So we can download the data sets or we can even use it in online and we can analyze the results in both these databases. Other than this, what else did we discuss? Other than network analyzed SGNet, impact very good. So the one thing is impact and the Chinese database, Hub 2.0, equivalent to impact. We have lot of Chinese hubs, isn't it? So whatever we consume the chocolates, whatever is available, the chocolates available in our markets, we don't find any herbs, isn't it? But here in the China, so many chocolates they incorporate herbs, different types of herbs. Okay, so they are utilizing the traditional medicine very effectively. Chinese people are using effectively compared to Indians. Okay, so this is something about the network for pharmacology, and also we have discussed on a particular vaccine, Zolgensma, gene targeted delivery. This vaccine is produced by Novartis for what disease? Zolgensma vaccine for what disease? I told you, in 2018 itself, it cost around 18 crores. Now it's more than 25 crores. A single shot of the vaccine. What is it used for? Very good. Spinal muscular atrophy. Okay. Now let me go ahead with the today's session. Okay. So today we'll be discussing something new. We start with the vaccine design. So till now, whatever discussed is related to drug design. Now, let us come to this immunoinformatics and reverse vaccinology in internship and certificate course in AI and ML approaches of drug and vaccine design. So, whatever earlier we have seen, we can call it as a pharmacoinformatics. And we can also have something called bioinformatics. The bioinformatics is if we study informatics in biological molecules, we call it as bioinformatics. But if you use informatics in immunology, we call this as immunoinformatics. So today's contents we discussed on immunoinformatics, vaccine adjuvants, types of vaccines, reverse vaccinology, case study in reverse vaccinology, indigenous database, that is whatever database we developed in our institution, epitope databases like B cell and T cell. Now let me go ahead with immunoinformatics. What is immunoinformatics? This we also call it as computational immunology, which is a field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. And if you see this computational immunology, majorly here we study this immunoinformatics to come up with discovery of vaccine as well as vaccine adjuvants. What is vaccine? Vaccine is a biological preparation that induces both acquired immunity and, of course, humoral immunity. The acquired immunity, whatever I'm telling, it consists of both cellular as well as humoral. Both the immunity, we simply call it as acquired immunity against a particular pathogen. For example, if you want to get a SARS-CoV-2 vaccine, so we have to take the SARS-CoV-2 components, isn't it? Only these components can uh, use to prepare the vaccine for SARS-CoV-2, and this can't be used for SARS-CoV-1 or MERS-CoV, the other kind of coronaviruses. Okay, whatever virus we take that as an agent to prepare the vaccines, isn't it? So vaccine requires adjuvant. This is an additional material particle. So this adjuvant is used to stimulate an effective immune response. So in my lab, we are working on adjuvants for the past 15 years. That is in Department of Biotechnology, SVC. And my mentor is Professor M. Sivanandam. He is former director of New York Medical College and St. Vincent hospitals in USA. So he worked in adjuvants for the past 40 years, I believe. Okay, so he worked for around 40 years. He has vast experience in the USA itself. He also brought patent in the melanoma vaccine, USA patent in melanoma vaccine. So I'm his student. An adjuvant is a chemical or biological agent that is added to a vaccine to boost the humoral or cellular immunity by increasing the efficacy of antigen. So whatever adjuvant we take, okay, it can be a chemical or biological. So 100 years before, the most popular used adjuvant, even nowadays, if you see Pasteur Institute of India, Konur, okay, so this Pasteur Institute of India, Konur, is majorly manufacturing what kind of vaccine? What is a major product of or major vaccine Pasteur Institute of Konur is manufacturing? The Pasteur of Institute of India is present in so many European countries. Very good. TT, DT, diphtheria. The most famous, which is circulated all over India is the rabies vaccine. Okay, of course, even whatever the vaccines you mentioned is also there, but rabies vaccine is an exclusive, is an highlight of the Pasteur Institute of India, Konur. Actually, they are using alum as an adjuvant in the rabies vaccine. And biological adjuvant agents. Our lab is working on different biological agents. In the end of this presentation, you can see what are the biological adjuvants we are manufacturing. I mean, we are using even for in vitro and in vivo studies. And if you see this humoral, here I mean acquiring the immunity through antibodies. Acquiring the immunity through antibodies. Cellular The cellular immunity can be acquired through APCs. APC, it means that antigen presenting cells. The antigen presenting cells, the examples, if you see, B cells, dendritic cells, macrophages. Okay. So these are examples of antigen presenting cells, natural killer cells, etc. So if these cells, whatever I mentioned, this will be interacting with T cells, T lymphocyte cells. If it interacts, you can see the T cell gets activated, and few cytokines, if interact with these cells, even these cells gets activated. So what is the purpose of activation? The purpose of activation is to even proliferate the cells. One cell is proliferate into two cells. So similarly, you can find the cells will be multiplied, and even the proteins produced from the cells are getting multiplied. Again, I'm telling you, from this chapter, we should have a knowledge on immunology. Okay, earlier there is no need of immunology, just like that. If you know the drug information related to pharmaceutics, pharmacology, that is sufficient. Okay, so also you should learn immunology to learn about immunoinformatics. Now we can find this kind of an applications of adjuvants. Now let me come across these types of vaccines. So before going to the immunoinformatics, which we find majorly reverse vaccinology. So before going to reverse vaccinology, first we should know what is vaccinology. So for that vaccinology, I just illustrating with the types of vaccines and with the corresponding examples. Inactivated vaccines: hepatitis A, flu, polio, rabies. Live attenuated vaccines: measles, mumps, rubella, smallpox, chickenpox. Before going to the other vaccines, can someone distinguish or the difference between? Tell me the difference between inactivated vaccines and live attenuated vaccines. So, can someone unmute and answer for this? This we are studying from school level, isn't it? There is no difficulty. Yeah, tell me.
Yes, sir, I can answer that.
Yes, sir, please, sir.
So it is a inactivated means using some chemicals or process we can make it inactivated like formaldehyde, so it won't replicate. Whereas a live attenuated is a continuous attenuation form, but still in the live form. So in such a way that the pathogenicity is reduced.
Very good, sir. Excellent. So actually, what we do here, first I tell about the inactivated vaccines. We can use heat, we can use chemicals like formaldehyde, and we can make the viruses as inactivated. That is, whatever viruses we have, this won't have that active nature. If it is inactive, the inactive viruses can be used to kill the active viruses. That's what we call it as inactivated vaccines. And in live attenuated, okay, we remove the virulence factors, whatever the proteins responsible for that viral which gives a viral response, that will be, so it will be the viruses are live, not killed like inactivated vaccines. These are live, but only the virulence alone is removed. So this won't, this kind of viruses, whatever we have, this can also can target the active viruses. Okay. And if you see the examples of the measles, mumps, rubella, nowadays you can see MMR vaccine, which can target all these three. Okay, so multi-component kind of a vaccines, and smallpox, chickenpox, and messenger mRNA vaccines. I think many of them know about the COVID-19 vaccines. Best example for messenger RNA. Subunit vaccines or protein, a part of a protein. Like if virus is getting inside our cell, even our cell has a component called proteosomes. The name itself called proteosomes. Proteosomes are majorly responsible for degrading proteins. If virus is entering into our immune cells, the immune cells has the proteosomes which degrade that viral membrane and releases the proteins. And whatever proteins we have, okay, even it can further degrade into peptides. Okay, so these kind of a small unit of proteins or peptides we call this as subunits, and recombinant and polysaccharide, okay, and conjugate. So this also comes under one category. Meningococcal disease. The meningococcal disease, the popular thing is the bacterial. This is caused by bacterial meningitis. There is one bacteria called bacterial meningitis, which can cause us this meningitis. Okay, meningitis. Okay, this causes this meningococcal disease, usually occur in brain, and HPV stands for the human papilloma virus. I've already mentioned that 99 percentage of the cervical, I mean 99 percentage of HPV infections definitely causes cervical cancer. Okay, so cervical cancer is majorly associated with these kind of viral infections. This is what we call it as comorbidity of diseases. Okay, so for this, you can use these kind of a components instead of taking the killed viruses or live attenuated viruses. We can use the components for treatment, and not for treatment, for prevention of disease. Vaccines, drugs. What is the purpose of vaccines? To prevent upcoming of disease. Okay, to we acquire the immunity, isn't it? So only we go with these kind of vaccines. Toxoid vaccines, the toxins produced from the bacteria. Toxins produced from the bacteria can be used to kill the bacteria, like diphtheria, tetanus. Okay, now tetanus virus. They not just up to the bacteria, even the viruses can also produce the toxins. And viral vector vaccines, COVID-19. Okay, the popular thing, Covishield. Remember, we have a DNA adenovirus vectors. Okay, that we used in this COVID-19 for treatment of COVID-19. I mean, for the prevention of COVID-19 infections. Peptide-based vaccines. The best is Covax. More about this we'll be discussing. Covax is a kind of a multi-tube peptide-based vaccine. Reverse vaccinology. Just now, whatever I told about studying about the vaccines, how the vaccines are prepared directly from the viruses, bacteria, and parasites. Okay, can be killed or attenuated stage, or even the part of components can also act as a component for preparing the vaccines. And of course, if you want to produce a major production in pilot scale, we need a bioreactors to produce that. Covishield and Covaxin also produce like that. Okay, and if you see this reverse vaccinology, this is majorly a computational part, a computational informatics. Okay, computational immunology or immunoinformatics, majorly we use this reverse vaccinology, where you should have, we start with the existing data like a genome, proteome. Okay, in genome we can see the genes which can code for the proteins, and the proteins you can see even our PPA interactions can also give some knowledge on that. So whatever we have the genome, proteomes from the proteome databases, and we link with the structures, we find the structures from that. Okay, and from that you can able to generate the peptides. If you generate numerous peptides, we screen the peptides based on a methodology, and then we can come up with a test peptide which can treat a specific population. Using this reverse vaccinology, we can treat cancer, viral diseases, bacterial diseases, also autoimmune diseases. We can treat any of the diseases using this reverse vaccinology approach. And we can also able to precisely treat any disease. For example, if you want to treat human, the Tamil Nadu Mudalia or the Andhra Chettiar, or I want to treat, so I can able to target even up to the community. That's what I'm mentioning. If you want to treat a state-wise population, or you want to treat even a community population, so whatever vaccine we can develop, that is going to target specifically 100% you can cure when compared to the others. Whatever we vaccine we develop for the Indians might not fit for the Americans, might not fit for the Africans, but we have can also design vaccines for that specific population also. Okay, so more about this we'll be seeing in the subsequent slides. Now about reverse vaccinology. I told you earlier, we start with the genome sequence, proteome sequence, we go with the different analysis. We have an exclusive tools and databases for immunoinformatics. Whatever we used initially for the drug design, we are not using here. We use exclusive tools and databases here. The most popular, widely used database is IEDB, Epitope Database. But we have hundreds of databases like this. So whatever we want to use, we can use the databases and we can use the corresponding tools for mining the data. And whatever we get thousands of data, we screen the best kind of a epitope. Epitope, I mean peptide. A peptide epitope is our basic output. This is what we have to generate. Earlier in drug design, the output what we generate is a chemical molecule, a drug molecule. It can be a synthetic molecule or it can be even from natural sources. But here, our objective is to generate this peptide epitope. The size will be around 8 to 15 amino acids. This 8 to 15 amino acid peptide can able to cure the specific diseases, whatever we have targeted. Okay, so for this, if you see, obviously we start with the genome proteome and then we try to find the specific antigen for that. For example, if I go with SARS-CoV-2, what is the most important protein in the SARS-CoV-2? We have 20 proteins, I told you. We have 4 structural proteins and we have 16 non-structural proteins. The most important is a structural protein called spike glycoprotein. The Covishield and Covaxin are all relying on with the spike glycoprotein. Only through the spike glycoprotein we come up with these kind of vaccines, isn't it? So if we have to take such a kind of a spike glycoprotein antigen, and from this we can able to come up with the kind of a discovery of B and T cell epitopes. Okay, whatever we discover, we can try with the in vitro studies, and we can also later try with the in vivo experiments with small animals, and then we can try with the healthy volunteers. Okay, so if you want to study about the epidemiology, we can study the epidemiology patterns, and we can screen the best epitope according to the epidemiology. Like I told you, the Indians, whatever the vaccine design for Indians definitely won't fit for the Africans and Americans. Then we have to design for them specifically. Okay, so considering the molecular epidemiology, if we go with a kind of appropriate peptide-based epitopes, and first initially we try with healthy volunteers of around 100 and above 1,000. We go with phase two. So whatever we try or healthy individuals, and after trying with healthy individuals, then we have to try with the disease volunteers. However, we work with drug design, even for the vaccine design, the similar kind of studies are done. See, in March 2019 and around April or March 2019, the world is discovered as pandemic. Okay, epidemic to pandemic. The epidemic we find in March, and I think in April they declared as pandemic. So within that April 2019, you see, April 2019 to September 2020, around 89 vaccines are developed. How many know? I don't know. Anyone know? about the Covishield and Covaxin, and we got around in 2021, I believe. So it is came to market in September 2020, but the government issued later in the end of 2020 and 2021. Other than that, Covishield and Covaxin, we also have 87 types of vaccines generated. So all this or cleared these kind of a trials. Okay, up to at least phase two and a part of phase three. If this is not coming up to clearing up to the phase one, phase two, and a part of phase three, these vaccines are not in the market. Okay, now let me go ahead with this reverse vaccine. And even these vaccines, we can able to take it for safety, and we can, if it is safety, we can go with FDA approval, that is post-marketing surveillance, which comes under phase four kind of a trials. And if it is recommended, it can be used. Now let me come up with an example for the reverse vaccinology. So this is a GAT question. What is the first vaccine discovered from reverse vaccinology? In the world, the first vaccine discovered is meningococcus B vaccine. It's a meningococcus B polysaccharide kind of a vaccines. This is used for treatment of bacterial meningitis, the bacteria which affect the brain. So how this kind of a vaccine is discovered, initially, if we see the proteome kind of a databases linked with the structural data, protein databases, we come up with around 600 potential vaccine candidates. And from 600 potential vaccine candidates, we use a strategy that how many proteins can be successfully expressed in E. coli. Because for a small scale, no problem. But if you want to treat a major population, we have to go with recombinant production. So if you see out of 600 potential vaccines, only 350 proteins only successfully cleared expressing in E. coli. Later, the next step we go with how many or surface receptors. If we identify only 91 novel proteins are identified from 350. And the next step of what we have done, what they have done is they check for the bactericidal activity. I told you this meningococcal B vaccine is used for treating bacterial meningitis. So for bactericidal activity, only 28 candidates screened, and this screened candidates is what used to come up with epitopes, to come up with various epitopes. Thousands of epitopes are there, but the best epitopes are used for the treatment of this disease. Now let me come up with a case study. Okay, the case study, what I'm going to explain is the publication, what I done in this pandemic time. Actually, this I completed before the pandemic time, but it has taken some time for publication. I worked with two type of epitope based vaccine design, B cell as well as T cell. But the B cell epitope based vaccine design was already published. And for the Nipah virus, the best vaccine design is the B cell. But for few viruses, the best to work is the T cell epitope. Okay, that depends on the types of viruses. Some viruses are RNA viruses, some viruses are DNA viruses, some are single stranded, double stranded, multiple. So we have types of kind of viruses. Okay, so based on the kind of viruses, we have to check. But I tried with both. But for a publication, I just published the T cell based epitope. Actually, when I started working with Nipah virus, what first motivated me to work on this Nipah virus, in 2018, this Nipah virus affected a particular group of population in the state. Which state is that? In 2018, are you studying the news? In 2018, one state alone got affected with this Nipah viral infections. And even, we have a movie, a famous movie called Virus. The virus movie is related to the Nipah virus. I don't know how many I've seen, but I'm just asking you, what is that state? Which state is getting more affected because of viruses? This is one more clue. Kerala. You forgot, I think. Okay, Kerala got affected. Okay, and nearly 18 people died because of Nipah virus. The one thing in the Nipah virus, unlike SARS-CoV-2, SARS-CoV-2 we have mutations, 100 types of mutations, and the hundreds type of strains are there. Fortunately, for Nipah virus is not going to get mutated, so we have only one strain, but that is deadly. Okay, so who are getting Nipah virus infections, they are isolated in the hills, and no treatment is given for them. Just like that, they have to die because no treatment is available. So this made me to think about and work on this kind of a strategy using a reverse vaccinology. When I started working, whatever structural proteins and unstructural, non-structural proteins present in Nipah virus, all the data are already published at that time. I have only one option for me to work with a protein called W protein. W is a part of a structural protein of Nipah virus. This alone didn't work. Of course, this is also a virulence protein. So I started working on this kind of a protein. And whatever I worked, this is also got funded from TNSST, that is from Tamil Nadu government UG projects. And also this selected as best project in national level, in national award. Okay. So we worked with the UG students worked up to that. And what exactly we worked, I'm showing you. Initially, we have taken the Nipah virus protein W sequence, and whatever we have taken, uh, we identify the T cell based epitopes. Okay, and after finding thousands of epitopes, we screen the best epitope, and whatever we screen the best epitope for the specific Indian population. So in this case, if I screened for the Kerala population, whatever I screened, I just we have to talk with the human HLA protein. Human HLA. HLA stands for human leukocyte antigen. Human leukocyte antigen is a gene. It's a type of a gene which codes for MHC molecule, MHC proteins. Actually, HLA we call it as a gene. This code for MHC protein. MHC stands for Major Histocompatibility Complex. Major Histocompatibility Complex. This is present in antigen presenting cells. This is a surface receptor of antigen presenting cells. So, what I did is we discover an epitope from a virus. In this case, is Nipah virus W, and we try to talk with the surface receptor of human HLA molecule, that is MHC protein. Then, after finding the binding patterns and energy score from the software, we finally go with the molecular simulation analysis to check the stability. To check the binding affinity, we go with docking, and to check the stability, we go with the molecular simulation. So, about the next kind of a virus. So, still we have plenty of opportunities to work with the discovery of vaccines with SARS-CoV-2. The Covishield and Covaxin, whatever we have tried, it can give efficacy only best efficacy only to the first strain. For the remaining 99 strains, we have to design a separate vaccines. Covishield, Covaxin doesn't work for the other strains. Even if it works, the efficacy will be very, very less. So my next question is, what is the maximum efficacy of Covishield and Covaxin? So we have taken Covishield and Covaxin. We are in Indians. We are living in India. I think you know about these vaccines. So whatever data reported in 2020 and 21, okay, what is the efficacy reported? Because only the 2020 to 21 we find the first strain. At 21 to 22 we can see multiple strains came into existence: alpha, beta, gamma, delta, iota. So many kind of strains are available. Very good try. And then any other answers you can answer for this. First I asked about the Covishield. Now I'm asking also the Covaxin. Which gives a better efficacy, Covishield or Covaxin for the Indian population? Good. So one already tried Kerala. Okay, Covishield. No, actually, The Covishield, the overall data, like based on the population, how much they have tried. The Covishield came around 83%, but could try. And then Covaxin came around 91 to 92%. So even if you take 92%, so this is the maximum efficacy. That means the Covaxin can able to prevent 92 people from 100 people. The 8 people we can't able to prevent because of variation in HLA genes. Each and every human distinguishes in fingerprint. So only we have fingerprint sensor, isn't it? The employees use the fingerprint sensor for the attendance. Similarly, if we see for you and me, the hemoglobin protein sequence is same. Myoglobin protein sequence is same. Insulin is also same. But there are a specific proteins which distinguishes between us. Not only between us, even if we are siblings, okay, even the siblings have a distinguished HLA genes. Okay, like however we find the fingerprint, even the HLA genes for the siblings vary. So then one can able to see that only that HLA genes can give you an knowledge on whether that vaccines is going to prevent or not. So this is actually membrane, nucleic acid, and proteases, polymerase. The types of proteases, polymerase considered non-structural proteins. And if you see a kind of T helper cell epitope, okay, that one universal T helper cell epitope is tagged with five specific cytotoxic lymphocyte and T helper cell epitopes. Okay, and this is actually 1 plus 5 is six peptides are tagged now. So whatever is tagged with each other, this is tagged with a TH1 CpG. Okay, and then it is stacked with an adjuvant, that is an aluminum phosphate. And in this side you can see this is stacked with a receptor binding domain of spike glycoprotein. Okay, so this kind of a construct 1 plus 6 plus that is 7 plus 8 plus 9. So this 9 in a single construct. So whatever that's what we call this as a multi-toe based vaccine. You know, the efficacy of this vaccine, the efficacy of this vaccine reported is more than 99%. More than 99%. And this vaccine, no one heard. This was used only in US. The US people has prepared this kind of a vaccine, and this used in US and also demonstrated in Taiwan. Now let me go with this COVAX vaccines. COVAX is of course a product of reverse vaccinology, immunoinformatics. With respect to safety, excellent profile when compared to the monoclonal antibodies as well as small molecules. Okay, and efficacy, we get a good kind of efficacy reported there. It is specific and targeted, and when compared to this monoclonal antibodies, this Covax is somewhat cheaper. Okay, this is cost effective, expensive. Okay, and this is stable over a period of time. So there are multiple beneficial effects with the multi-toe based vaccine. So only we are discussing on these kind of vaccines. Okay, that is peptide based epitope vaccines. The first type is the T cell epitope based vaccine. So in T cell epitope based vaccine, okay, we have this MHC2. Actually, this is a small change, MHC2. So we have T cell, CD4 T cell. CD4 T cell stands for what T cell? Cytotoxic T cell. See, if you are working with the MHC2, we call this as helper T cell. This is actually helper T cell. That is, the helper T cell has CD4 molecule on the surface. And CD8 T cell we call it as cytotoxic T cell. So T subscript C, we call it as cytotoxic T cell. Okay. which has CD8 on the surface. So this is how the T lymphocytes are distinguished. Helper T cell has CD4, cytotoxic has CD8, but you don't have CD4. Okay, now let me come across. Other than the CD4, what else? So if the CD4, we can see a T cell receptor. Okay, these two molecules are going to interact with MHC2 molecules of APC. There is one surface receptor called MSC is interacting with CD4 as well as T cell receptor. What is the T cell receptor? This is what shown as the T cell receptor. Okay. So what is the purpose of interacting with CD4 and T cell receptor with MHC2 molecule? So just because of this interaction, the T helper cell gets activated. So one T cell is going to get proliferate into two T cells. The T cells can combat immunity. I mean, can provide immunity or combat the cancer or viral antigen, viral particles. Okay, so if you want to combat the type of diseases, of course, we need more T cells and more cytokines released from this interaction. Cytokines or the soluble proteins like interleukins, interleukins are the type of proteins we call it as soluble cytokines. This, if it is released from the cells, these can directly combat along with tumor necrosis factor proteins. There are numerous immune proteins which can also combat the virus, also the cancer. The tumor necrosis factor is a popular proteins, is a group of proteins which combat cancer. TNF, in the TNF, not all the TNF molecules, specifically tumor necrosis factor alpha. There are some kind of proteins called interferons. We are even in interferons we have alpha, beta, gamma, but interferon gamma specifically can combat viruses. If we find more interferon gamma, we won't get these kind of viral infections. Like some still didn't get any chicken pox. If someone not get infected from chicken pox yet, so they have more interferon gamma. Okay, that is understood generally. So like this, if you see the cytotoxic T cells, which has CD8 on the surfaces, along with the T cell receptors, okay, interact with the MHC1 molecule. So antigen presenting cell has MHC1 or MHC2, or it can have both also. Some APC has both MHC1 and MHC2 on the surfaces. But the APC of MHC1 interact only with the CD8 and T cell of cytotoxic T cell. If it interacts, obviously cytotoxic T cell gets activated. Again, same kind of proliferation happen. One T cell gets proliferated, and even the cytokines release. If it interacts with the other receptors of APC, even APC gets activated. So APC also gets multiplied. Now let me come across with the next kind of an epitope, that is B cell epitope. So till now we discussed about types of T cell, that is TH and TC epitope. Now we are discussing the types of B cell epitopes. Here also we have two types. Okay, here if you see in B cell, the B cells are the most important lymphocytes which can provide us a specific immunity called humoral immunity. Humoral immunity happens only through antibodies. So the providing immunity through antibodies can only be generated from the B cells, B lymphocytes. Here, if you see these two cases, this is one type of B cell epitope and this is another kind of an epitope. If you see the first type of epitope, we call this as continuous residues, linear epitope. And if you see the second thing, discontinuous residues. Why we have discontinuous residues? Why we have continuous residues? See this example. We have have amino acids in equal distance. So only call this as continuous residue. So only we call this as linear epitope. Why we have discontinuous? Because one of the amino acid is getting inside the antibody. So this length is more, this length is somewhat less, and the next length is more less. So in this case, we need an epitope sequence. The epitope sequence is needed. Epitope, I told you, around 8 to 15 amino acid. But in this case, if you want to design, we need epitope structure. That if only if you have a 3D structure of an epitope, we can come to know how effectively it is going to bind. So whatever you see here, this is what the antibody binding region. The antibody binding region, we call this as paratope. And whatever we see this epitope, okay, this is a sequence from the virus. If it we are coming up with the Nipaviral W protein, okay, then the Nipah from the Nipavirus W protein, we get a part of a peptide, okay, which is going to bind with the human antibody. So the antibody contacting region is paratope, and that viral contacting region, that peptide, we call it as epitope. So there are two types of regions, epitope and paratope. Okay, and if you see the previous slide, okay, so one thing we have to observe, whatever we see this peptide, this is what we have to discover, design. Okay, so whatever we get this peptide is from the viral source. If it is Nipah virus W, this is Nipah virus W peptide. And if you see this helper T cell, just because our peptide is having this kind of a conformation, that is going from outside to get inside and coming outside. So this is generally binding with the MHC molecules. Whatever Nipah virus W, okay, generally what it happens, the Nipah virus is going to engulfed by antigen presenting cell. Like macrophages, this directly engulf the viruses, can be Nipah virus also. Whatever the viruses it is taken inside, okay, these are processed. I mean, initially through proteosomes, this Nipah virus is going to degrade, and uh, whatever proteins we have, these are degraded into peptides. A part of a peptide is going to bind with the MLC molecule, and the MLC molecule is projected on the surface. So this is what we call it as a presenting and processing. Okay, so I mean processing and presentation. So whatever we present the peptides on the surface of the MHC2 molecules, this gets interacting with the T cells. And if you interact with the corresponding receptors, the T cells gets activated. Okay, and whatever T cells gets activated, get a memory to attack that kind of a Nipah viruses. So whatever peptides we have, that is specific to that viruses. If a person is infected from SARS-CoV-2, the SARS-CoV-2 peptide is projected at the surface. And whatever the T cell going to get interacting with the APC, the T cell is going to get that acquired immunity, a kind of a cellular. This is what we call it as a cellular immunity. A cell and cell is going to interact, and a type of activation what occurred is a kind of a cellular immunity response. And so whatever the activation happened, the one T cell is going to proliferate into two T cells. The T cells can recognize that SARS-CoV-2 viruses coming inside, and this directly go on combat. And even the molecules released, that is especially interferon gamma released during this activation, can also combat directly that SARS-CoV-2 viruses. Okay, like this, proteins and cells together can function effectively. And here in this B cell epitope, however we find that antigens, okay, the viral antigens, like however we find the viruses, if even if we work on cancer, we find the cancer antigens, the abnormal cell proliferation happening, target proteins. We go with it, and we can also come up with cancer vaccines. This concept we can use to come up with a cancer based epitope vaccines. Okay, so do you understand the difference between this linear epitope? In linear epitope, we need the sequence, just the peptide sequence. So even that we find it in the form of continuous residue, so on we call it as linear. But in the second type of basal epitope, we should have an epitope structure. If you see the epitope structure, that epitope is going to bind effectively, the antigen is going to bind effectively with the antibody. So we can able to find the discontinuous residues. So only we call this as conformational related to structure. Okay, so whatever you design the conformational epitopes or more precise, this is more precise when compared to linear epitopes. So linear epitope is just an informatics data, but this is more than the informatics data. Even if you check affinity purification, antigen and antibody has more than 99% affinity when compared to any other molecules, a small molecules, even peptide-based interaction or even small molecule interaction. The antigen antibody is the best kind of an affinity. It shows the best. So obviously, the structure of antigen and structure of antibody thing we are checking here. Now let me go ahead with this receptors. So like T cell receptor, even we have B cell receptor that is having this kind of a structure. Okay, more about the T cell and B cell receptors and that kind of an epitopes we'll be discussing during the next sessions. Helper T cell, cytotoxic T cell, you remember? So just now I explained you. Okay. So why just because our antigen is coming out and in, this size of this helper T cell epitope is large. So we can fix the length up to 13 amino acid. So many try even with up to 15 amino acid. So 13 to 15 is okay, but I suggest to go with the 13 amino acid. And for cytotoxic T lymphocytes, just because the interacting patterns, if you see in the MHC2, we have two different macroglobulin proteins. Whatever we find beta 1 and beta alpha 1, these are two different macroglobulin proteins. Just because of two different macroglobulin proteins, we have to design a kind of a large peptide covering from outside to inside. But here, in this MHC1 structure, you can able to see alpha 1 and alpha 2 or the subunits of one macroglobulin. This is not a two different macroglobulin, only one macroglobulin, but these are two subunits. So there is no need to come up with an extensive, lengthy peptide. So here I suggest you to go with 9 amino acid, but the preferred range you can take is 8 to 10 amino acid. So if you want to do your research, you can go with 9 m. Fix a particular length and go. That is easier for to complete this research. Okay. So actually, whatever I worked with the Nipah virus W protein, whatever I completed the computational studies, immediately that after the publication, a PhD scholar from Pasteur Institute of India has taken this kind of a research to complete in vitro studies. So whatever you do the computational studies, definitely it helps others to start from this that whatever we have completed to complete in vitro and in vivo studies. So even generate these kind of a data, definitely it gives a sense to come up these kind of a vaccines for them. Now let me go ahead with the dendritic cell markers. Of course, this is one of the professional antigen presenting cell. So even we can come up with a discovery of vaccines respective to the cell. We have T cell based vaccines, dendritic cell based vaccines. So for coming up with this dendritic cell based vaccines, T cell based vaccines, we should understand about the cell and the contacting receptor, surface receptors. Myself, in my PhD research, I worked on all the 162 surface receptors of dendritic cells. Okay. So I worked with all the surface 162 surface receptors of dendritic cells, and I also, along with the PG students, we have come up with a kind of a tool for treatment of autoimmune as well as cancer diseases. Like generally, we come up with different molecules, plant-based molecules, and which plant molecule is best for the treatment of any specific disease that we can able to make it. This dendritic cell-based vaccines can be used for treatment of different types of cancers and different types of this viral immunity. It can also combat the viruses. Then, uh, what surface receptor is responsible, and even what kind of phytochemical interacting with what surface receptor. So this is of course an important to come up with a kind of interaction studies for this. Whatever plant molecules are interacted, this can be used as an adjuvants. So more about this, if I see in the subsequent slides, you can understand. So one more thing you have to understand, that I told you something about T cell activation. What is the T cell activation? If dendritic cell receptor of MHC2 or MHC1, if it interact with the CD8 or CD4, this alone won't make T cell gets activated. One more important thing is that this is of course most important receptor. Along with this receptor activation, we should also have co-stimulatory marker like CD80 or CD86. CD80 or 86, if it interacts with the CD28, okay, the T cell gets activated. But instead of CTA28, if it interacts with the CTLA4, cytotoxic T lymphocyte antigen 4, the T cell gets inhibited. So T cell activation is helpful for treatment of cancer. T cell inhibition can be used for the treatment of autoimmune diseases. That the treatment of cancer and treatment of autoimmune disease is quite opposite with each other. If you have more T cells, we can combat cancer. If you have less T cells, then we can combat immune autoimmune diseases. Example, if you see arthritis is an autoimmune disease. What is arthritis? A pain, wherever you find joint pains, that's what we call it as arthritis. There are 100 types of arthritis. The popular thing is rheumatoid arthritis. Around 26 joints, or we find this arthritis. Okay, so in that pain, the joint, okay, if you have more number of immune cells and more amount of tumor necrosis factor, this can also aggravate the progression of the disease or this autoimmune disease. So we should try to inhibit the T cell activation so that even this production of TNF etc. will be reduced, and so we can combat. Okay, so this is something about the, and the father of dendritic cell, Ralph Steinman, received Nobel Prize in 2014. And whatever I mentioned, treatment of autoimmune diseases through this co-stimulatory markers interacting with CDLA4. 4. I think this kind of a novel approach got a Nobel Prize in 2019. So, and we have similar kind of researchers are also awarded Nobel Prizes. Okay, of course, this is an important part of a discovery. So I just need 2 or 3 minutes to complete this session. So if you see if Nipah virus or any SARS-CoV-2, if it is coming inside, okay, just by phagocytosis by antigen presenting cells like B cell, dendritic cell, this can act, the dendritic cell macrophages or the best cells to engulf the kind of bacteria or viruses. If it takes inside, after getting into the proteasome, it degrades into peptides. These are transported through transported associated protein in the endoplasmic reticulum, and whatever peptides it's going to bind with the MHC molecules, and these MHC molecules are transported through Golgi apparatus, and from Golgi apparatus it's going to move through the secretory vesicles, and finally these are exported on the surface of the antigen presenting cell. So whatever that small antigen, okay, you can find on the surface of the APC is going to interact with the T cell receptors, and obviously T cell receptor activation proliferation happens. Okay, this is an example of MHC1 processing followed by presentation. The final thing is the presenting on the surface. The processing happens in the immune cells, that APC immune cells. So MSC1, MSC2 structure, I told you MSC1 is a single subunit, but MSC2 you have two different subunits. So we need a large peptide for MSC2. And if you see that MHC1, MHC2, okay, these are the most important genes. The most important genes, in MHC1 are A, B, and C. Okay, if you take a kind of a chromosome, so what is this chromosome? Chromosome 6. Chromosome 6 has ABC, that is which code for class 1 MHC, and it also has the TP, TQ, and DR. So these are the important genes. Okay, and if you want to discover the epitopes, the whatever we have discovered the epitopes, we have to target these kind of genes to check the immunity. There are also class 3 MHC, which is less significant when compared to MHC1 and MHC2. So this is used for complement system discovery. Okay, more about the difference between MSC molecules. We can study this from immunology textbooks by Reut and Mail, QB, etc. Okay, so this is something about immunology. So other than the major MHC class molecules, anyhow, tomorrow we'll be seeing more hands-on session. Before going to the hands-on session, we should know minimum immunology. Without minimal immunology, if I start going explaining how to use the tools and databases, there is no significance. You don't know how to infer the results also. So one should come up with this immunology. So a major MHC class 1, you see ABC, and major MHC class 2, you find TPDQDR, which code for okay, so the MHC class 2. And other than this, you can also find minor EFG. So very rarely we work with this minor MSc class 1 and MSc class 2. So in the chromosome 6, where exactly HLA is coded on our B and C, you can able to find it from the short term or the large term. Most important website which we are going to explore on tomorrow is the IEDB. Tomorrow we work with the T cell epitope based vaccine. In the IEDB website, you can see both the T cell epitope prediction tools, B cell epitope prediction tools. We also have databases. In the databases, we have 9.88 lakh structure peptides are already discovered. This is not updated up to present. We can find many epitopes, peptide epitopes or non-peptide epitopes are deposited in the database. So we are using the tools, and we are mining the data from the databases, and we show the results. Just we have to work in online. And related to historical development of vaccine adjuvants, our lab worked extensively on the usage of IL-2 adjuvant, IL-4, interleukin-2, interleukin-4, granulocyte macrophage colony stimulating factor adjuvant. So whatever immune molecules we use, they can also used as an adjuvant. AH03, which uses plant molecule saponin as an adjuvant. Okay, we are come up with discovery of few more plant molecules that can act like an adjuvant. What is an adjuvant? This is used as a component to increase the efficacy of antigen. If you want to have a prolonging of immunity in our body, we can add these plant molecules along with the vaccine to increase the efficacy. And even dendritic cells can also used as an adjuvant. So our lab worked extensively to develop dendritic cells by treating with adjuvants, and even whatever we can use, we can also use it for dendritic cell therapy for treatment of types of cancers. We have whatever the database we have developed. I'm showing it to you. This is we named as an immunoanalytical database. We have developed it from C Sharp, SQL, and PHP. Like this, we can come up with any kind of a strategy. And this is an action adjuvant database, plant-based adjuvant database. So using this plant-based adjuvants, you can able to screen the specific secondary metabolite, okay, for the treatment of a specific disease. For arthritis, which is the best plant compound. For the asthma, which is the best plant compound. Okay, like this, for each and every disease, we can come up with a vaccine, and the best adjuvant can also be found out from the tools. Okay, so this is a tool we have developed in our institution. So more about the T cell based vaccines and B cell based vaccine, especially with respect to hands-on, we'll be discussing it on tomorrow. Today, I have completed the basic information of immunoinformatics with respect to reverse vaccinology. So, any doubts you can ask me. So, if no doubts, we can uh, discuss. If no doubts, we'll be seeing the session on the next session. Have a great day to you all. Bye. Thank you for your interactions and patience.